Good day, everyone, and thank you for joining us at this 2021 ceremony of TNQ's Inspiring Science Awards. This is the fifth year of these annual awards for the best paper in the life sciences published from India in the previous year. They aim to recognize and reward quality science, to inspire scholarship, and to encourage students of science to pursue excellence and do significant and creative work. The Inspiring Science Awards have steadily gained recognition in the Indian science community, and the applications we receive are of impressive scope, spread, and quality. This year, we received 554 entries from over 240 institutions across 100 cities in India. I would like to thank our jury who worked in 3 panels and spent the last 2 months assessing these entries to shortlist this year's 6 excellent finalists. Each year, applications are invited from young scientists who are registered for a PhD up to those in the 4th year of their postdoctoral research training. For the 2021 award, the research paper must have been first published either online or in print between November 1, 2019 and September 30, 2020. We intend to extend this period to a 12-month one from October 1 of the previous year to September 30 of the current year. The submissions should be full-length research papers that have been accepted and published. The author must have been affiliated to and the research conducted at a research institution or university based in India. Papers with co-authors will be considered, but the award will be given only to the submitting author. All 6 shortlisted finalists will receive an ISA medal, a citation, and a MacBook Air. The winner will, in addition, this year receive a cash award of 2 lakhs. This cash award takes a temporary pandemic place of an all expenses paid travel grant to a life science conference of the winner's choice anywhere in the world. We hope to reintroduce the travel grant as soon as possible. Anuranjan Anand proposed these awards as an extension of our annual TNQ Distinguished Lectures in the Life Sciences and as a way for us to engage with the research community through the year. He is Professor of the Molecular Biology and Genetics Unit and Associate Faculty and Chair at the Neuroscience Unit of the Jawaharlal Nehru Center for Advanced Scientific Research in Bangalore. Professor Anand has agreed to announce this year's winner and to discuss the 6 shortlisted papers with the finalists. They are Santosh Kumar Kuncha, a Senior Research Associate at CCMB, the Center of Cellular and Molecular Biology, Hyderabad. Richa Mishra, a postdoctoral fellow at the Laboratory of Microbiology and Microtechnology, Lausanne. The research for her paper was conducted at the Indian Institute of Science, Bangalore. Aarush Mohit Mittal, who is a doctoral student at IIT Kanpur. Ritika Tiwari, a postdoctoral fellow at the Cancer Biology Department, Lerner Research Institute, Cleveland, whose research was done at IIT Kanpur. Soumik Ray, a doctoral scholar at the Department of Biosciences and Bioengineering, IIT Bombay. Raj Kumar Verma, a senior research fellow at the Laboratory of Plant Microbe Interaction at the Center of DNA's Fingerprinting and Diagnostics, Hyderabad. Congratulations to all 6 of you. May I now invite Anuranjan Anand to discuss the work of our finalists with each of them and then announce the winner of the 2021 Inspiring Science Award, Professor Anuranjan Anand.
Thank you, Mariam. Warm welcome to everybody attending this ceremony. We have come together to felicitate the finalists and announce the winner of ISA 2021. I'm told that today's event is being attended by members of the research and academic community. Many of the institute members from where our finalists come from, families and friends of the finalists, ISA winners of the previous years, interested members of the public, colleagues from the TNQ. Thank you everyone for joining this ceremony. On one hand, this event is unlike previous years, where we are missing out on the distinguished lectures, and from the students and finalists' point of view, the whole excitement of an auditorium experience and thrill of getting to know that you are the one of the finalists or the winner. There are reasons, obvious reasons that everybody knows about it. And we also know that from now on, things can only get better. On the other hand, for the ISA 2021, this year has been much like the previous years, except a bit better. For the number of entries received, 550 plus, as Mariam indicated. From the number of organizations that participated, 240 plus. That's hugely encouraging. As you know, these awards aim to recognize and reward quality science to inspire scholarship and encourage students of life sciences to pursue excellence and carry out creative work and publish that. Going forward, the number of entries mentioned may just continue to increase, which is a testimony to the presence of a vibrant research community in biological sciences working on many aspects and flavors of the subject, which is something becoming evident in the range of papers which get submitted in these previous years, is what I'm told. Each year, applications are received from young scientists in the country. The process of submission is simple. I'm unsure if it can be made simpler. It needs no recommendation, no nomination, no forward letter. One has to simply go forward and upload or submit what one considers is the best published paper while working in an institute in the country during PhD or early stages of postdoctoral training. The idea of this award was proposed over 5 years ago to create a connect between the students and young researchers beyond the TNQ Distinguished Lecture series. The award is largely aimed at PhD students in the country or young researchers. I'm excited about this fact because I get to interact a lot with PhD students. PhD is perhaps the longest jump in the growth process of one's scientific journey. And whether one realizes it or not at early stages, before one reaches a level, for example, to submit an article for the ISA award, as this process unfolds, it tests one's enthusiasm, passion, motivation on one hand, ability to think logically, scientific reason, many experimental skills, knowledge of several techniques and methods, time management skills, abilities to organize, make presentations, collaborate, face rejections of papers, enjoy acceptance of papers, which is sort of very, very exciting time for any scientist. And these attributes stay with one and in fact grow no matter what one takes up as a profession subsequently. So PhD studentship indeed equips no matter what one does subsequently with many high level skills crucial in professional life. And by employing these in right manner, one can create a win-win situation wherein one almost never fails in life in a larger sense of the word. Just have a look at the value and the role of a modified PCR technique in these previous months. And one gets to learn many more methods during PhD or postdoctoral training. TNQ's fondness to connect, TNQ's desire to connect with the students by way of the lectures and ISA is well known. But just in case someone is unaware, I shall reveal that it took no more than a minute or so for Mariam to get convinced about the idea of initiating ISA and continuing commitment of the TNQ team to nurture it, to think of ways to expand its reach in the community of young researchers and students in the country. Today, we have an opportunity to join with the finalists, discuss about their work with each one of them in the presence of many eminent members of this scientific community, friends and families of the finalists, and my congratulations to each one of them. I request all of you to put your hands together to congratulate all our 6 finalists before we move on to invite Santosh Kumar Kuncha.
Hello.
Okay, so we
warm good evening to all of you.
All
Am I
right.
audible?
So
Yeah.
yes, you are very much audible. So let's. I would like to begin this process with Santosh Kumar Kuncha, who is currently a senior research associate at the Center for Cellular and Molecular Biology, Hyderabad. Santosh, what I need to know from you is, while you are currently at the CCMB, um, where did you do your master's and undergraduation?
So I have done my master's from JNTU, Hyderabad, Jawaharlal Nehru Technological University, whereas my undergraduation from Loyola Academy, Alwal, that's in Hyderabad itself.
And when did you join CCMB?
I joined the CCME in 2014 for PhD program, and I have recently submitted and defended my viva in the last month.
Wonderful, wonderful.
Yeah, yeah.
So, Santosh Kuncha has published a very nice paper in eLife. I'm not sure how many of people have seen your article. I'm sure almost all the members attending this function and ceremony today. And your paper deals with an unexpected result from the genome sequencing projects.
Yes.
Given that I sort of think a lot about the human genome, I'm told there are about 300 or so ISO decoders in human genome, and I'm sure there are many more in animal kingdom.
Yes.
What about plant kingdom? These decoders are not in the plant kingdom. Is that the
That's
idea?
right. No, even plants have many ISO decoders, but the functional characterizations of the plant thing is still going on. So that has to be looked at. So it's just an emerging field with respect to ISO decoders. This term itself, we just come to know. A few years back, this term ISO decoders has been introduced, and the work is just started to begin. So earlier, if you look at bacteria, it just has around 50 tRNA, or 40 to 50. If you look at, as you have rightly stated, humans are around 300 ISO decoders. But if you look at the numbers, also even the numbers, it's nearly 600 tRNAs in humans. But plants are no exceptions to this. With the increase in the genome size, there is an increase in tRNA numbers also. But however, the functions of these are just coming up. So recently, there are many articles which talk about tRNA fragments, role of tRNA in many other conditions such as cancer, ribosome biogenesis, regulation, epigenetics. So there's much more to it. The field is just coming up.
Right. So the title of Santosh's paper is Genomic Innovation of ATD Alleviates Mistranslation Associated with Multicellularity in Animalia. You indicate that tRNA expansion of decoders is what you mean, has led to misselection resulting in critical error of alanine mischarged onto tRNA threonine, which needs to be proofread by an enzyme called animalia-specific tRNA deacylase ATD in vitro. You've gone on to show that in addition to ATD, there's a threonine tRNA synthetase, THR-RS, which can clear the errors in cellular scenario. And then you mentioned about two-tier functional redundancy for the translational quality control or surveillance. Would you elaborate? And then you tie in very interestingly story of reactive oxygen species, etc. So how is all this put together? I have read some of your additional articles on the subject where you mentioned that ATD is a paralog of DTD. Are there more players like ATD and THR-RS which are likely to be present? So I found this sort of very interesting and fascinating area of work. And I would like that you elaborate in about 2 minutes time a bit more about this before we move on.
Yeah. So as rightly mentioned, the title itself tells that you have a new enzyme to take care of a mistranslation, which is induced because of oxidative stress. If you move from a single cell organism to a multicellular organism, the functionality of the cell increases. And hence the ROS production also increases. It's not that ROS is bad. ROS is actually used as a signaling molecules in case of multicellular organisms. It's well characterized as well. But excess ROS is deleterious to the cell. But when we identified this protein, what is we came across this increases in number of tRNAs in animalia. And if you look at, there are few tRNAs which are charged with alanine, which are supposed to be charged with threonine. So wrongly acylated tRNA. So tRNA is selected wrongly here. And this will lead a mistranslation at the proteome level. So this needs to be corrected. Just like our DNA replication, we have proofreading domains. Similarly, we have proofreading enzymes working at different stages. We are talking about the translation. And this tRNA, we wanted to understand how it works. When we initially did a knockout of ATD, we realized that the cells are happy. But the moment we started giving ROS from external source, then the cell started showing phenotypic effects because of mistranslation. So then that struck us the idea there has to be something in the cell which is sensitive to ROS. And that is where we discovered another enzyme, that is threonine tRNA synthetase. And we realized that even that can edit. So earlier reports have never spoken about threonine tRNA synthetase editing L-alanine. It is always shown to be edit L-serine. So it was totally unexpected in that time. But we could show both in vitro and in vivo that threonine tRNA synthetase, when it is active, it actively deacylates alanine, which is mischarged on tRNA threonine. Therefore, it does not allow threonine to alanine mistranslation at the cell. So if you look at, there is a functional redundancy, which is, so during oxidative stress, one of the protein is suppressed. Basically, it's inactive. The editing function is inactive. So you need another protein which takes care of this. If you look at the recent advances in tRNA biology, so one of the questions which we believe is very, very important and not yet addressed is how are tRNAs routed from translation to non-translation function, away from translation. So having
right. So Santosh, that's really very interesting. What I'm going to do is have you in JNC at some time point and have all our students listen to you. We need to move on. Thank you so much for sharing this very fascinating work with us, and congratulations to you.
Thank you. Thanks a lot for it.
So now I want to invite finalist Richa Mishra. Richa has published a paper in Science Translational Medicine. Hi, Richa. How are you?
Hi, everyone. I'm good. How are you?
Very fine, thanks. The title of her paper is Targeting Redox Heterogeneity to Counteract Drug Tolerance in Replicating Mycobacterium Tuberculosis. Richa Mishra has done her PhD working in the Indian Institute of Science, Bangalore. Before joining IAC, where were you, Richa, for your master's and undergrad, before we move on to ask you about your work?
I did my master's from the Department of Microbiology at the University of Pune, and I did my bachelor's in microbiology at St. Xavier's College in Calcutta, which is my hometown.
Okay. So Richa, your paper deals with a sort of very important problem. An unusually long-term treatment is required to cure TB in humans is known to all of us. And this is necessary to prevent relapses due to genetically drug-sensitive bacteria that becomes transiently resisted inside the host cells and tissues. And you have gone on to examine mechanistic basis of this in a very intriguing way to me. You have exploited heterogeneity, redox heterogeneity, which seems like a very subtle phenotype, biochemical, physiological phenotype, and have gone on to do a wonderful omics work, which has revealed involvement of iron sulfur cluster biogenesis, hydrogen sulfide gas, drug efflux pumps, and antibiotic resistance. Would you tell something more about how this idea struck to you? What was sort of interesting while you were thinking about and developing this question, that this is what you want to address? Because I felt sort of very excited about just reading the title and after that, many aspects of your paper.
TB has been a really tricky disease to cure even after 100 years of discovery of the bacterium that causes it. And a very important phenomenon to consider for this is multi-drug tolerance, where bacteria transiently are able to tolerate high concentrations of different classes of antibiotics. It's interesting to see that there are reports which show that this happens specifically when the bacterium is residing within its host in macrophages or in other immune cells in the host. And this leads to the generation of many different subpopulations within one single bacterial population that infects an individual. So what we're essentially looking at is heterogeneity in bacterial fractions within a single host or within a single immune cell. So this work was done with the redox biology group where I did my PhD under Professor Amit Singh's mentorship. And this has been a question that the lab has been trying to address for some time now. And in this regard, the group had developed a biosensor, which was specific to the redox state of Mycobacterium tuberculosis, which is the causative agent of TB. Now, it's important to look at the redox state of bacteria because a lot of the stresses that this bacterium faces when it's engulfed by, say, a macrophage in the human lung are all radical in nature. You have low pH, you have generation of reactive oxygen and nitrogen species. And so all of this have a very direct consequence on how it reprograms its redox metabolism, because that does allow it to survive for very long, even through therapy, even through exposure to different antibiotics. So the group has developed this biosensor, a redox biosensor, that helps us non-invasively quantify the redox state of bacteria in real time while it's within its host cell. And what we were trying to understand is how we could use this biosensor to probably characterize phenotypic heterogeneity that could lead to multi-drug tolerance. And we discovered that once inside the host cell, like a macrophage, bacteria very rapidly diversify into different fractions, some of which are more tolerant to multi-class antibiotic exposure than others. And what we wanted to do with this finding was to characterize how these different subpopulations are different from each other, what allows one bacterial fraction to be more tolerant simply by readjusting its redox state under certain conditions of stress like host residence or exposure to antibiotics.
So it's a very subtle phenotype in some sense. It's
Yes,
not a very dramatic redox potential differences. I was looking at the numbers that you have indicated. They sort of differed marginally. And
yes,
that makes so much of difference and has implications on downstream pathways.
right.
Really wonderful. Are you continuing on the same line of work in Lausanne?
So what I'm doing here is, since we talked about phenotypic heterogeneity just now and how different conditions within the host can lead to the generation of different subpopulations or bacteria that differ from each other, even though they are all genetically identical, what we are trying to do here is try to understand how drug action differs at a single cell level. So if you have a population of 100 bacteria, we believe that not all bacteria in that 100 genetically identical population would react similarly to antibiotic exposure. And we're using microfluidics and time-lapse imaging in real time to look at how single cells in a bacterial population can differ in the way they take up, react to, and take care of antibiotic stresses during infection to help
That's
make
wonderful.
therapy more
Thank
effective.
you. Thank you. Thank you, Richa, and congratulations to you.
Thank you.
Okay, let's move on to invite Ayush Mohit Mittal.
Yeah. Hello, sir.
Hi, Ayush. Are
Hi.
you with us? Wonderful.
Hi. Yeah. Hi.
Now,
How are
Ayush, I'm doing fine. Ayush, you are working in Department of Biological Sciences and Bioengineering,
Yes.
Indian Institute of Technology, Kanpur, in
Yes.
Nitin Gupta's lab. And
Yeah.
you published a paper in Nature Communications, multi-network properties overcome random connectivity to enable stereotypic sensory responses.
Yes.
Very, very nice piece of work. If I have to reflect on this a bit, what I know is that stereotypic behaviors, they sort of have evolutionary advantage.
Yes.
I have studied them in context of courtship, sexual activities, etc., in insects, and they can increase fitness and speed. And in that context, fixed action pattern of neurons is thought to be produced by a hardwired neural
Yes. Exactly.
network in response to stimulus. Neurons in some areas show random connections and in other areas fixed connections. And you seem to propose that can the brain regions which receive inputs from non-stereotypic connections can generate stereotypic responses. That's
Exactly.
something very intriguing. So how does it happen? I mean, you say, is your red same as my red? Or is your fragrance same as my fragrance, whichever one
Yeah.
you
Yeah.
would you tell us a bit about how did you go about raising this question, addressing this question? You're working
Yeah.
on a phase of locusts and flies. And in particular, tell us a bit about what is PRED, a new matrix you have evolved, and how it is
Yeah.
better than others. You have about 2 minutes to tell us about your exciting
Yeah.
work.
So yeah, it's a lot to tell, but so yeah. So the basic question we started with was when we looked at the insect olfactory system, which is the system that is involved in smelling different odors. So within that, in Drosophila and locust, and even in other insects, there is a multi-layered system of neurons that is present there. And it was, and in Drosophila, it is known that there is one layer of neuron that connects randomly to the next layer of neurons. So by that we mean that if we have two individuals of the same species, they will have very different connections. But just after the second layer of neurons is another layer, and its responses were measured in locus, and they were found to be very similar in different individuals. So that formed the basis of our hypothesis that how did random connections lead to very similar responses? And we went about it by using simulations and a mathematical analysis of the circuit. So we created the
Yes.
circuit within using code and also using mathematical principles. And we found that it is based. So the 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 layer that receives random corrections. So it preserves the information about the different odors within the layer itself. So uh, the entire population of cells comes together to store the information, and that is how they can preserve the odor information within the next layer. So that is one of the results, and there are other properties like the non-linearity of the input that the layer receives. And so neurons are non-linear in their behavior. So whatever input they receive, uh, uh, using different kinds of connections, so they do not do a direct linear transformation of that. So based on that, those are some of the properties that help the neurons to preserve this information and pass it on to the next layer. And the second part is about PRED. So PRED is a metric that we evolved to capture uh, the stereotypy in the system. So generally people use correlation. So correlation between two different vectors is used, and correlation by itself mostly captures how the two different vectors behave. So if one vector is increasing and the other vector is also increasing in the same way, then the correlation will be positive. If the other vector is not increasing, then the correlation will be zero, or it could be negative also, right? But it does not really capture the absolute values of the response. So let's say we have uh, so by absolute values, I mean that whatever is the spiking response of a neuron, that is the absolute value it can capture, right? So correlation kind of ignores that. So if we have two vectors like 1, 2 is one vector and 2, 4 is the other vector, then the correlation will be 1. And for 1, 2 and 3, 6 also, the correlation will still be 1, right? So it cannot really capture that change in the absolute value of the second vector. But
Right.
PRED,
Yes, I mean, I could appreciate the strength of your method as
yeah,
compared to the previous methods, and it's come out very well in your writing.
yeah,
So many congratulations to you, Ayush. Wonderful
thank you,
job. Thank you very much for joining in, and
thank
all
you,
the
sir.
best.
Thank
We need to move on. I would like to invite Soumik Ray. Soumik, would you please
Hi, sir.
join us? Soumik has published, where are you, Soumik? Soumik has published a paper in Nature Chemistry, alpha-synuclein aggregation nucleates through liquid-liquid phase separation. Wonderful piece of work involving 22 authors from 4 departments and organizations.
Right.
In itself is terrific. So congratulations
Thanks so
for
much.
participating in this larger collaboration and making things happen. My perspective of alpha-synuclein is from a very genetic point of view.
Right.
As you would know that this gene was found in kindred from Contrucci thermae in Italy.
Right.
In early 90s, this work began, and I think around 97 or 8, the gene was found. And discovery of this gene was a very powerful story in human disease gene identification, to the level that this discovery was mentioned by President Clinton in his 1998 State of Union address. It was a pre-genome era. Chromosome 4 had another important gene of Huntington, which was found by the work of several scientists working from 84 to 93. And then about 9 years after that, the gene for Parkinson's came in. Now, it's a story which has continued for almost 25 years or so. And you have added a very unique biochemical, biophysical angle, bringing in liquid-liquid phase separation to address early events of the aggregate formation. And you have used wonderful techniques, in vitro experiments, cell biology, bringing in mutations, all put together in one paper. Would you tell us in about 2 minutes and disentangle this for us to some extent?
Sure, sir. So we know that synuclein aggregates and form amyloid fibrils, which is the higher order structures of this protein is very toxic to the cells, which leads to neuronal cell death. But we didn't know that what triggers or what initiates the process of this aggregation, how in a cell or in a test tube, these synuclein molecules can come together to form higher order structures. So this is what we exactly show in the paper, that liquid-liquid demixing or liquid-liquid phase separation can be one of the initial events that brings all of these synuclein molecules together in a very tight space. And in that membraneless tight compartment, which is a droplet, essentially, the concentration of the synuclein molecules reaches to that extent so that it can nucleate the aggregation process. And we see that exactly that is what happens. Synuclein can form aggregates inside these droplets. And very interestingly, the factors that promote the aggregation of synuclein or PD, Parkinson's disease symptoms in general, they kind of also accelerates the process of phase separation of synuclein as well. And dopamine, as we all know, that dopamine is used as a therapy against Parkinsonism.
Right.
So that has shown to be inhibiting the phase separation process as well. So there's a direct correlation between the liquid-liquid phase separation and alpha-synuclein aggregation. That is what we exactly find out. And also in detail, we show that at a single droplet resolution, from inside the droplet, we have taken the anisotropium and the rigidity of the molecules, and we have done FRAP experiments to show that the translational as well as the rotational dynamics of these molecules inside these droplets slowly become slower and slower because they are aggregating inside. So they are becoming more and more restricted. And also we show in real time that how the amyloid fibrils are actually present inside these droplets by various techniques. And we have used, as you mentioned, that we have used mutations, the mutations that hastens the process of aggregation also hastens phase separation and otherwise.
So if synuclein has propensity to form aggregates,
yes,
why it does not happen in the wild type or a normal situation in a cell? Or does it happen, but it's not harmful, not so to the level that is harmful?
There are several hypotheses. One of the very popular hypotheses is you will get Parkinson's if you live for 100 years. A synuclein will eventually aggregate, but just that the time is different for different individuals. But another hypothesis is, yes, the endogenous expressed synuclein levels are so low that they cannot form nucleus until there is a redox stress or there is something wrong with the cells already. So there has to be a predisposition. There has to be, some reports suggest that there can be metal toxicity, which can act as a predisposition behind Parkinsonism. That leads to, that is exactly we show in our paper as well, that copper and ferric ions, these transient metal ions, they induce ROS in turn, that ROS kind of makes these condensates formation inside HeLa cells.
Yes, thanks. I read your paper with great interest. Congratulations.
Thanks so much, sir.
Thanks for joining. Let's move on to invite Ritika Tiwari.
Hello, sir. Hello, all the
Hey,
finalists, and
Ritika.
congratulations to all of you guys.
Yes, congratulations, Ritika, to you too. You have published a paper in Nature Communications. Androgen deprivation upregulates SPINK1 expression and potentiates cellular plasticity in prostate cancer. Now, I see that you have sort of tried to sort out a genetic regulatory conundrum involving SPINK1, REST protein, SOX2, all these names are mentioned in your paper. And you address the interplay of these in context of a special type of prostate cancer. All right. Now, I'm aware of the fact that prostate cancer is caused by one of these very curious tempress ERG fusion, and a large number of patients are due to that chromosomal, I think, translocation event, etc., etc. Where do the kind of patients you are looking at fit in, and how did you come up with this gene regulatory cascade and interplay of that, and what is its value in context of the treatment?
So, if I talk about the TMPRSS to ERK gene fusion, it is the most recurrent gene fusion in when we talk in terms of prostate cancer, and it is like present in like 50 to 60% of the prostate cancer patients.
Really?
However, however, there are other subtypes, like the second most lethal subtype that would be called as SPINK1 overexpression, which is a serine protease inhibitor casal type 1 protein, which is like overexpressed in 10 to 25% of the prostate cancer patients. And it is associated very adverse symptoms like early biochemical recurrence or increased metastasis, etc. So we are all aware that AR, that is androgen receptor, is primarily the governing factor or the driving factor for TMPRSS to ERK fusion. And it was also been established in the literature that TMPRSS to ERK fusion is actually mutually exclusive to SPINK1 overexpression subtype. So the question, it came to us naturally that if AR is actually driving TMPRSS to ERK fusion and SPINK1 is overexpression is mutually exclusive to TMPRSS to ERK fusion, then what is the status of AR signaling, and what is the correlation of androgen receptor signaling in context of SPINK1 overexpression? And this kind of led the whole story unfolded it in front of us. Then since since AR is the common driving factor in TMPRSS to ERK fusion in many other prostate cancer subtypes, what happens is that ADT, that is androgen deprivation therapy, is sort of used for the treatment. However, we found out that AR, that is in androgen receptor, actually negatively drives SPINK1 expression. And we found out that if we are giving androgen deprivation therapy to the patients who are actually SPINK1 positive in nature, what would happen is actually it will elevate that repression that SPINK1 had, and leading to overexpression of further increased expression of SPINK1. And this increased expression of SPINK1 actually promotes cancer progression rather than actually suppressing it. So the conventional therapy, which is good for which might serve the purpose in other subtypes, might not hold true for in context of SPINK1 positive prostate cancer cases. And another thing with the other thing that we identified was that AR, along with its co-repressor REST, actually are driving this process of repression. And further, we also identified that if the patients are given ADT, it leads to the development of a lethal subtypes neuroendocrine prostate cancer. And we found out that through patient studies as well as through cell lines and publicly available omics data, we were able to identify that SPINK1 actually correlates with this neuroendocrine phenotype, which is in fact very difficult to treat, and there is no therapy as such for that. And to conclude our study, we were able to identify one of the small molecular inhibitors against CK1 protein, which actually stabilizes the REST protein, and that might be used as a therapy and as an alternate therapy for SPINK1 positive prostate cancer patients instead of conventional ADT.
But you have participated in a large collaborative project, almost 15 authors, many institutions. It's a really privilege. It's really privileged to get involved in such work. because many of the future studies in biomedical areas, the kind of thing perhaps you like, are going to be of that nature. So wonderful training, wonderful piece of work. Many congratulations, Ritika.
Thank you so much, sir. Thank you.
Thanks. Let us move on to invite Raj Kumar Verma.
Hi, Professor.
Hi, Raj Varma. Raj Varma is a student at Center for DNA Fingerprinting and Diagnostics. Raj, where were you before joining CDFD?
I did my master's in Tezpur University, Assam.
Okay.
And I came here.
Your undergraduation in Assam too?
No, I am from Rajasthan, so I did my undergraduation over there. LBS College, Kotputli, it's near Jaipur.
Interesting. So I think education lets us travel and see a lot of interesting places. Now, you have published a paper in Cell Reports. A bacteriophytochrome mediates interplay between light sensing and the second messenger cyclic GIGMP to control social behavior and virulence. You indicate that light is one of the most important environmental signals and is sensed by diverse forms, but many attributes of this aren't very well understood. And you reflect that bacteriophytochromes are the most abundant and ubiquitous light sensing receptors in bacteria and involved in time of the day behavior or responses. That's very versatile. However, their biological and regulatory role in non-photosynthetic bacteria is poorly understood, and less is known about how they regulate diverse cellular processes. You go on to study ZooBHPH, very interesting abbreviation. Xanthomonas oryzae bacteriophytochrome from this plant pathogen, which causes bacterial leaf blight of rice and perhaps of other plants, as you indicated. Now, would you tell us, you've carried out elaborate piece of work, but we want you to give us a sort of slice or a flavor of that, telling us about virulence program that is regulated molecularly, how it is associated with the functions. And you have carried out a genome-wide expression analysis of XOBPHP regulon and reached very specific answers from that kind of omics study, which isn't easy actually. So we want to hear from you something about this work.
So in general, bacteriophytochromes are having two modules. One is sensory module, which is made of PAS, GAP, and Phi domain. And the other one is response regulator, which is in general in phytochromes is histidine kinase. But this phytochrome from the XOO doesn't have that response regulator. Instead, it is having PAS domain, which is in general involved in the sensory things. So this makes it's interesting. So what is the downstream signaling for this bacteria after light sensing? So we started looking at its protein structure because that is the only thing which can give us hint about the functionality of the protein. So we found that gap domains from the literature, that gap domains are all involved in the cyclic NMP or the NMP levels of cellular modulation, which in turn binds to different response regulator and function according to the signal they receive. So in that case, we started looking what kind of NMPs it can modulate. So we have done the non-specific substrate using paraphospho, BIS-BP, and we have done the PDA phosphodiesterase activity assay, and we got a signal that it's actually having a phosphodiesterase activity. Then we started looking which one is the specific substrate for it. So we have tried with different these nucleotides, and we got that it's a cyclic di-GMP specific phosphodiesterase. Then we started looking, okay, it's sensing the light signal and it is having phosphodiesterase activity. What is the physiological relevance for the bacteria after sensing the light? How it is helping the bacteria in its physiological activities? So this bacteria naturally enters in its host by hydrothodes, which are xylem openings. Okay, so bacteria is always on the epiphyte or on the philosphere. It has to find these hydrothodes and it has to enter. So for that, it needs some signal. So in that, we have proved from our paper that in its natural mode of enter, it takes the signal and it modulates the cyclic diGMP level in turns. Those levels will change the virulence associated functions, and they will guide the bacteria or the timing of bacteria to enter into the host. So this is what we have overall revealed from our work.
Thank you, Raj. Congratulations to you, the team, and the lab. Really enjoyed your coming here and sharing your work.
Yes.
Many congratulations. We need to move on to the next step in the process, and that has to do with announcement of the winner. In spite of rather close competition, one of the papers of one of you stood apart according to all the jury members. That work is tour de force at the level it has been done. And that individual has gone after an important problem in a very bold manner, attempting to address key questions about biochemical, physiological, and molecular attributes of the process. A number of different experiments masterfully executed, starting with confocal and flow cytometry to categorize the macrophages on the basis of subtle redox potential phenotypes, and going on to address the key attributes to molecular level, which reflect on Mycobacterium capacity to tolerate the drugs. I think is no secret in the manner I have gone to describe. Our winner is Richa Mishra. Let's congratulate Richa Mishra on this achievement. It's a major achievement going by the fact that we had 540 plus entries. 6 of you are all winners in some sense because it's a very, very small percentage. And it's been challenged for nearly 30 plus jury members to select 6 finalists from across the regions. But Richa Mishra's work stands out. So very many congratulations to you, Richa. And I'll stop here and hand over the process to the TNQ members. See, Richa's work has established a link between phagosomal pH, redox metabolism, and drug tolerance in replicating MTB, and suggested repositioning of chloroquine to shorten TB therapy and achieve a relapse-free cure. Richa, hello.
Hello. Sorry, Anuranjan. I'm delighted to welcome Dr. Soumya Swaminathan, our surprise special guest at the ceremony. Dr. Swaminathan, as you will all know, is Chief Scientist of the World Health Organization at Geneva and a widely cited expert on various aspects of COVID-19. Soumya Swaminathan is a clinician and a pediatrician by training and a globally recognized researcher on tuberculosis and HIV. She spent most of her career as a researcher and then as director of what was earlier known as the Tuberculosis Research Center in Chennai, TRC. It is now called the National Institute for Research on Tuberculosis. Before Dr. Swaminathan moved to the WHO in 2017, she was Director General of the ICMR and Secretary to the Government of India. A highlight of her leadership at ICMR was bringing scientific evidence to the formulation of health policy and the development of research capacity in medical schools in India. Soumya Swaminathan has published more than 350 peer-reviewed articles and book chapters. She's an elected foreign fellow of the US National Academy of Medicine and a fellow of all 3 science academies in India. It is apt and an honor that Dr. Soumya Swaminathan has agreed to interact with our 2021 Inspiring Science winner, Richa Mishra, and to discuss her paper, Targeting Redox Heterogeneity to Counteract Drug Tolerance and Replicating Mycobacterium Tuberculosis. Thank you, Dr. Swaminathan, for being with us today. May I now invite our winner, Richa Mishra, back to join a discussion with you. Dr. Soumya Swaminathan, joined by Richa Mishra.
Thank you, Mariam. First of all, congratulations, Richa.
Thank you, ma'am.
I understand that you're also in Switzerland, in Lausanne, up the lake, from Geneva, and hopefully pursuing your work on tuberculosis. I'd also like to congratulate all the others who were shortlisted and who were interacting earlier in the program today. And it's clear that, to be 6 out of 500 young scientists selected as a finalist is wonderful. So congratulations to everyone, and thank you for your work, and hope that you will keep up with your own areas of interest that have brought you to this point. I think tuberculosis is very close to my heart, of course, because I have seen the impact that TB has on individuals, on families, and particularly on children who often get the most severe forms of TB, like TB meningitis and miliary and disseminated TB. And despite all of the advances over the last decades in better diagnostics and better treatments, which took a long, long time to develop, we still, face more than 10 million new cases and over 1.5 million deaths a year. So we see today with COVID that every death is tracked, every death causes moral outrage, and the world wants to come together to solve the problem. We've been able to develop vaccines within a year, and there are hundreds of trials going on around the world to develop new drugs and new vaccines for the SARS-CoV-2 virus. But that level of interest and funding and attention has not, unfortunately, TB has not had that. Maybe because it's an ancient disease, it's been with humankind for a very long time, and people just accept the fact. And I think also an important fact that it does affect the developing countries or the lower middle income and low income countries, and even within countries like India. It's clearly the poorer sections of society who bear a disproportionate burden of TB, even though it's an infectious disease, so theoretically infects everyone or anyone, but the risk factors and the determinants, the social and economic and environmental determinants are unbalanced, so they're borne much more by the poor. Now, coming to the paper, Richa, and I'll ask you to say more about that, because it's a very interesting exploration that you have done and on this concept of drug tolerance in Mycobacterium tuberculosis, which is relatively recent, and there's been so much work done, including by a good friend of mine, Dr. Lalita Ramakrishnan, who using the fish,
the zebrafish.
Yes, the zebrafish, thank you, model, has really gone in depth into the structure of a granuloma from the early stages of how granuloma forms, and has gone into a lot of the molecular mechanisms of happening within those granulomas. And that molecular understanding is important, because that's how you can then start thinking about how to target drugs at each of those different levels of different enzymatic pathways that get involved. Now, drug tolerance is an important, as you point out in your paper, mechanism for survival both of actively replicating and non-replicating Mycobacterium. And I think anything we can do to make the current regimens more effective. Current regimens last from 6 months to 24 months. And that's far too long. It's expensive, it's toxic, it's inconvenient. And therefore, the so-called host-directed therapies, which act through pathways within the human cells, in this case, macrophages, in a way that they facilitate the killing of mycobacteria is an important area to explore. So there have been many drugs that have been postulated to have an impact on drug tolerance. And I think the evidence you provide for chloroquine needs further exploration, definitely. I think the animal models seem to be promising, but we need to then take this work further and see whether it can go into the clinic. And that's where we need the linkage between the basic scientists and the clinical researchers. I think I'm not sure what you're doing at EPFL, you can tell us, but there's obviously a strong group there that works on TB as well. And so wish you all the best again. Congratulations. And I hand it over to you now to say a little bit about your work.
Thank you very much. As Dr. Swaminathan described, TB has been around for a very long time, and an absolute effective relapse-free cure still remains elusive for us. So I think the inspiration behind the paper, as Dr. Swaminathan mentioned, was when Professor Ramakrishnan showed how residence within the host can induce drug tolerance, and not just in non-replicating bacteria, but actively replicating Mycobacterium, which was digressing from the general dogma that was in the field, that only non-replicating bacteria can tolerate antibiotics because of a general shutdown of metabolic and physiological mechanisms. So that inspiring piece of work was very important for us to define our questions, to understand how actively replicating bacteria can diversify within the host to tolerate antibiotics. And I think it was not only important to discover or to look at these bacteria in these models, but also to identify what host cues are crucial to the generation of these drug tolerant bacteria. We were able to do so. We were able to define pH, acidification in the phagosomes, to be an early crucial cue that leads to the generation of drug-tolerant bacteria, some drug-tolerant bacteria inside macrophages. And the most important part of the paper, I think, is the fact that we can use chloroquine, which is an already approved, safe drug, already being used for the treatment of malaria in the field, to be used as a host-directed molecule that can sort of homogenize these different subpopulations inside the host to make them definitely more susceptible than they normally are to the multi-drug treatment that TB patients receive, because they're already given a combination of 4 antibiotics for 6 to 9 months in the simplest cases. So if addition of CQ to this combination can make a more efficacious regimen to not only reduce the bacterial burden within the hosts, but also to prevent any relapse or to generally shorten the duration and prevent the appearance of drug-resistant strains of TB, I think that could be a very important point to look into in the future to just make TB management more effective and relapse-free. Yeah.
Thank you, Richa. I'm sure you are aware of the department you graduated from has a long history of contributing to the basic research in the area of TB with Dr. T. Ramakrishnan, who's former professor and chair. So at various time points, various levels, that department has played sort of significant role in this research. So congratulations. As we come to the close of Inspiring Science Awards 2021 ceremony, may I thank Dr. Soumya Swaminathan, the Chief Scientist of the World Health Organization in Geneva, for taking time from her very busy schedule to grace this occasion with her presence, and for an illuminating discussion with Dr. Richa Mishra. Very many thanks to you, Dr. Soumya Swaminathan, for joining us.
Thank you, Anuranjan. Entirely my pleasure. And I would have loved to listen to all the other discussions as well that you had with the finalists. But yeah, it was a really matter of time But thank you so much for inviting me to share with Richa and to congratulate her. And I hope that this work can actually progress. Very often we see excellent work done by basic scientists, but that remains as a very good paper and doesn't progress. And we really need that translational part of research to become much stronger, I think, not only in India, but around the world. And we've been talking here about new models of drug development, where we don't wait for big pharma, to come in and pick up, but we really have a public-private partnership with the government, with academic agencies and labs and the private sector. And this is something that we should promote in India, because I think we have all the different components. It's a question of bringing them together to really focus on our own priorities and to try to bring science and link it to public health. Thank you so much. Bye.
Okay, so my warmest congratulations to everybody, to Richa Sharma. Wish you many more greater peaks of achievements in your career. And I stop here. Thank you.
Richa, would you like to say a few words?
Thank you. I'd like to share this with all the finalists where we are at. I'd like to thank the jury and TNQ Technologies for choosing our paper for this honor. To everyone who's worked on the paper, all my collaborators and colleagues, everyone at IISc, it was a great place to do your PhD. But most importantly, to my PhD mentor, Professor Amit Singh, who's like a constant source of inspiration in my career, and I probably wouldn't have done any of this without his support and guidance. And to my friends and family, thank you very much.
Thanks, Richa. Throw a party for your
Sure.
lab in IASC and in Lausanne.
Sure. Yes, once COVID's over.
That's true.
Great. All right. So as we come close to the Inspiring Science Awards 2021 ceremony, I would again want to thank Dr. Soumya Swaminathan, who took time from her busy schedule to grace this occasion with her presence. And thank you, Professor Anuranjan Anand. You are the architect of these awards, for being our chief guest and announcing the winner. And also thank you for the illuminating discussion you had with each one of our finalists. I also would like to thank all of the 32 judges, panel chairs, and jury members who took time to evaluate these papers, despite all their academic and research commitment, 500 or 550 odd papers. Application for the 2022 Inspiring Science Awards will open on October 1st this year. I expect we will receive even more applications this year, and we are starting to plan for that. I want to mention and thank two of our previous winners for joining us today. They are the Inspiring Science winner for 2020, Subhash Mehto, and the winner for 2018, Nishan Chettigar. A special mention to everyone at TNQ who worked very hard to make ISA 2021 possible. Kuri, Shankar, Praveen, Sri Ganesh, Swapnil, Kirtana, Sam, Vendan, and Shanti. Thank you, guys. On account of COVID-19 related travel and other restrictions, this award function has had to be online and could not be part of the 11th edition of our TNQ Distinguished Lectures in Life Sciences. Professor Jennifer Doudna of the University of California, Berkeley, was scheduled to be our featured speaker this year. As most of you know, she helped pioneer the study and use of CRISPR-Cas9 as a tool for gene editing and was awarded the 2020 Nobel Prize in Chemistry along with Professor Emmanuel Charpentier. Her much awaited set of lectures has now been rescheduled, and Professor Doudna will, we expect, present her lecture in Bangalore, Mumbai, and Delhi in January 2022. Today's event has been attended by close to 300 members of the research and academic community, many from the institutes that our finalists represent, family and friends of our finalists, interested members of the public, and my colleagues from TNQ. Thank you everyone for joining the ceremony. And congratulations once again to all of our finalists and the winner, Richa Mishra. I wish all of you great success in your scientific research career. All the best, stay safe and stay well. Thank you for joining.